Good afternoon. So today we are just going to look at the population structure and the plant demography. So these are the things that we are going to cover. First, we're going to look at density and pattern of distribution, then plant demography, and then under plant demography we're just going to look at a few things there, especially the population growth models and also the modular growth. Then we're going to look a little bit at the life tables and survivorship curves. Then the meta populations, we are going to look at them at the later stage. So the plant population, as you know, a plant population is just a group of plants of the same species which are seen to be occupying a particular area at the same time. So these are the components which are used to construct plant communities. So we have factors which influence the distribution of plants or plant populations. So the same factors influencing the presence of plants in some areas and also the absence of plants in some areas. So those factors include the environmental conditions, the resource availability, competition, disturbance, availability of propagules, or also termed the biogeography. And we have the measurements and distribution of plant population structure and dynamics. So distribution of plants on the landscape, so we'll be talking about the density and also the pattern of distribution. Then we look at the measurement of plant demography. So we'll be just looking at the changes in plant population size and also structure through time. Then the distribution of sorts of population within the landscape. Here now we'll be talking about the meta populations. The density, so the density is simply the number of individuals per unit area, which you should be familiar with from the previous classes. So it is most easily used for population of discrete individuals such as trees, and then less useful for species that reproduce vegetatively, such as grasses and other smaller trees. So we've got three ways in which plants can be distributed in nature. We've got random, clumped, and also regular. So they give more information on the population's habitat preferences, the competitive dynamics, the microhabitat distribution than the density alone. So in random distribution patterns, the location of one individual has no bearing on another individual's location. But in the clumped, you find, or in regular, you find that the presence of one plant may be affecting another plant which is nearby, possible through competition or allelopathy, or the clumped distribution of propagules which are from the parent plant. So a chi-square test can be used to test the random distributions. So these are the three ways in which plants can be distributed in nature. So when you are asked, you need to be able to remember these three and then illustrate them the way they are appearing and be able to tell us a few stories about them. So here we have an example of a clumped pattern. So this is due to the presence of some special microhabitats. So we have some environmental features. So it is said here on the barren land, we have high salt accumulation. So which means only species which are adapted to salinity can survive in those bare patches there. But as you can see, plants are trying to avoid these patches because of their high salinity, and then occupying areas with less salinity. So these are just some of the environmental factors which affect the distribution of plant species in nature. Then we come to plant demography, so which is just the study of populations, the change in population structure through time. So the plant populations increases or decreases through birth and also death or processes, as well as immigration and emigration, and by the vegetative sprouting. So it includes, so plant demography normally includes counting the number of genetically distinct individuals, the number of vegetatively produced individuals, the number of leaves, the branches, the tillers, the stems, and flowers, and so on and so forth. So the movement of seeds into and out of a population and storage in the soil. So we have to count that, all of those, if you are a plant demographer. And we have the idealized plant history. So now this is just when you are asked to explain, to talk about the plant demography, this is what you need to give to us. So as you can see there, we're starting off with a seed pool containing some seeds. For example, 26 seeds in the seed pool. And then out of those seeds, not all of them are going to germinate. So we've got the environment, which is going to act as a sieve. So it chooses which seeds are to germinate and which ones will not germinate. So this is because some of the seeds will fall on unfavorable environments or soils or on the rocks, and others will fall on favorable conditions, rich soils. Some might fall in waterlogged environment, which is also not favorable. So then out of all those that germinate, not all those that germinate are going to reach reproductive maturity. Some are going to die off. That's why you see we have seedling cores. So some are going to end up just there as seedling cores. And the other number might reach maturity, reproductive maturity, and some might even reproduce vegetative daughters. And therefore they can produce seeds which represents the next generation. And then, as you can see, this is a cycle. So these seeds are going to go back into the soil through what we call seed dispersal, and then form a new seed pool containing this large number of seeds. So you need to familiarize yourself with these numbers. So I'm going to send you some slides which are going to explain to you in details about what is going on with regard to this diagram. And we have different kinds of plants. So we've got those which are produced from a single genetic individual, which are known as genus, and then we have a group of distinct individuals that are part of a single genus, known as clones. And then we've got ramus, which is just a single member of a clone. It can be a branch of a genus. So plants have different growth from animals. Animals have determinate growth. For example, with humans, we only have one heart, 2 lungs, a liver, and 2 arms. and sometimes a determinate size. But with plants, they have indeterminate growth. That is, their size and abundance of parts can vary a lot because of different environmental conditions. So different number of shoots, leaves, roots, flowers, fruits, or seeds in response to unfavorable conditions. Similarly, their size can vary markedly depending on their location. In the desert, they might be very short. In the mesophytic conditions, they might be very tall. And then in the xeric environment, in the hydrophilic environment, they might have large leaves. So they may react to stress by varying number of parts. Then we have what we call the modular growth. So a concept of a plant being a population of parts. So from your roots, leaves, stems, flowers, fruits. So these are what we call modules. So plants may independently allocate growth to different modules depending on the availability of resources and also the environmental conditions. Then we have a concept which is known as fitness. So fitness, especially to animals, is known as the lifetime reproductive success of an organism. So this concept is much easier to apply to animals than to plants, because we are saying that if you are an animal and then you are unable to reproduce, then you are not fit. For you to be reproductive, to be biologically fit, you need to be able to reproduce offsprings which are going to represent you in the next generation. But with plants, their ability to reallocate reproductive efforts to vegetative modules during times of stress makes the analysis of fitness difficult without following the reproduction among all its vegetative shoots. Because when it can produce new shoots, then it is also known as reproduction. But it reproduces vegetatively. So that is why the field of plant demography is not that easy. Another important difference between plants and animals for population studies is the size distribution. So unlike animals, mature plant sizes are rarely distributed as normal curve, so in which most individuals are of moderate size. So plant sizes normally have an L-shaped frequency distribution of sizes. Furthermore, the size of plants is rarely a direct function of age. So you might find a plant being very short, but then being 10 years old. And then you might find a plant being very tall, but then you find that it only has 5 years of existence. So the size does not determine the age, but with humans, sometimes it might be easy. The age is also often not a good predictor of reproductive status in plants, because a plant can grow very fast depending on the conditions and also the type of plant it is. And then it can reach maturity at a very young age. For example, your invasive plants, they can reach maturity in a matter of few months and then be able to reproduce the seeds which will represent them in the next age. So this is how a graph of the plant looks like. So it's an L shape. what we call the population growth models, so which are used to predict the populations of plants at some future time. So we have what we call the discrete models. So based on Harper's diagram, we can derive the simple equation nt plus 1, which is equals to nt plus b minus d plus I minus e. So nt plus 1 is just the number of plants in some future time, and then nt is the number of plants at present. B is the number of plants which are being established from the seed pool, and then D is the number of plants that die off, and then I is the number of plants which are coming into the site, and then E is just the number of seeds which are emigrating from the site. So you take this and make it your equation 1. And then we have the continuous time of growth models. So these are said to be useful for predicting population at any time in the future. So you need to know the rate of population increase. So you're going to use R is equals to B minus D. So B is just the births and D is the number of deaths. So you take this and make it your equation 2. And then with R, we can then be able to calculate the rate of change in a population N. So you take this and make it your equation 3. And then you will see where we are going with this. So with that being said, then what is R for curve A? So that is if A is less than 0 or the other way. So we are going to see this, and then we have this graph. Therefore, we're saying that graph A is representing an exponential growth. So if R is greater than 0, the population will grow exponentially. But if R is equals to 0, then the rate of change in the population is 0. So which means that is a stable population. But then if R is less than 0, the population will decline. Therefore, what type of growth does B represent? So we're saying this is an exponential growth. Then we're seeing B, it has some limitation. So B there is said to be a logistic growth curve. So in this case, the population is constrained by some environmental limitation to a carrying capacity. So if the number of predators is too high, and then the number of prey is less. If the number of prey becomes less, and then the number of predators will also decrease. So they need to stay in balance. So the population will slow as it reaches K. So the equation for curve B is then dn over dt is equals to Rn into K minus n over K. This is what we call the Verhoeven's equation. So if you remember correctly, I told you that you need to write equation 1 from the equation wherein we are calculating the population size at some future time. We're saying nt plus 1 is equals to nt plus b minus d plus I. That's equation 1, and then we said R minus B, R is equal to B minus D as our equation 2, and then dn dt is equals to calculating the population, and then until we reach this equation we call the Pearl-Verhulst equation. So at some time you are going to be asked to derive with explanation of symbols this particular equation, which means you need to give a stepwise. You need to give a stepwise. So it's not actually a mathematical derivation as it is sometimes the way we ask it in the test or exams. So it's just a stepwise way in which we reach the Pearl-Verhulst equation, but not a derivation of it. Because if we try to derive this equation mathematically, it will take a different form. So your equation 1, you start with the equation 1, which is the one where you're calculating the population size at some future time, nt, and then you come with your equation 2, which is r is equals to b minus d, where you're calculating the rate. That is your equation number 2. And then you come to where you are calculating the population, because now you've got the rate, so you're going to use the n over dt is equals to rn, and then that is your third equation. And then finally you come to equation 4, which is the Harper's Pearl equation. And then we find that this Verhulst Pearl equation, it sometimes does not usually apply to the real world of plants, because the carrying capacities are really constant, so they vary with the environmental conditions. The birth and the death rates are also not constant, therefore R is not constant. The biomass of plants may have more impact on the carrying capacity than the number of individuals. Then the population boundaries are usually poorly defined. So these are the shortcomings of the equation. You are asked to give the shortcomings of the Verhulst Pearl equation. This is what you are supposed to list. These are the shortcomings or the disadvantages of the Verhulst Pearl equation. And then we have the survivorship curves, which you also need to be familiar with from your second year classes on Bio 2542. So type 1, these are the characteristics of organisms with the mortality rate concentrated in the later stage of their lives. So plants such as the annuals with seed dormancy. And then the type 2, the characteristics of organisms with constant mortality rates. And then type 3, the characteristics of organisms with high juvenile mortalities. For example, most of the trees. And then note that the large animals tend to have the type 1 survivorship, and also small animals tend to have the type 3, whereas the opposite is true of large and small plants. So plants, they are not like animals or humans. They have a different ways. Then we have what we call the fecundity and also the net reproductive rate. So you just need to read through these terminologies and then just familiarize yourself with those. And the same here, you just need to understand what is the relative value. So you're not going to be asked to calculate these ones. So if you have noted, I have been skipping some of the slides, which means you are expected to do the same. You can just ignore the slides which I skipped, which means they are not part and parcel of what you are supposed to cover at this moment. And then we're going to look at the meta populations. Then we are not going to explain them at this stage. I will explain them at the later stage. So this is just the summary of what we just covered. We are going to look at some mortality rates. So first we're just going to talk a little bit about Harper-Weinberg's law. So we're saying that he discovered that an equilibrium will arise and be maintained in both allelic and genotypic frequencies in any diploid population that is large in size, undergoes random mating, has no mutation or migration of individuals, and is subject to no selection. So the Harper-Weinberg law produces an equilibrium because no matter what is starting genotypic frequencies are, you will always come out with the same results. So the important conclusion is that the gene frequencies or genetic variability in a population will be maintained over time without change unless outside forces are applied. So evolution results from these forces. So these are just things that you have been exposed to since your first year cell biology and your second year also cell biology. So we are not going to concentrate much on this. So we're going to move quickly on to the more important stuff that I need you to understand. So these are just some of the factors which we were talking about: the mutation, the migration, which you are expected to go through them on your own, the population size, for mutation to occur, you need in small populations, chance may be critical, and then we give examples, and then random mating, natural selection. Then we have what we call the mortality rate. So you need to pay more attention to this now, because this is what we're going to be talking about, the instantaneous. So they are expressed as finite rates or instantaneous rates. And the number of deaths in a short interval of time is proportional to the total population size at that time. The rate of drop in the numbers can be described by a geometric equation: dn over dt is equals to in. If you can look closely, it's just the same equation which we talked about when you talk about the plant demography, the third equation. So where n is just the population size, I is just the instantaneous mortality rate, and t is the time. D is just the change. So in integral form, then we have nt is equals to no e exponential it for instantaneous rates, or nt over no is equals to e instantaneous rates, where no is just the starting population size and nt is the population size at time t. So when we're taking up the logs, if t is equals to 1 time unit, we obtain log e into nt over n0 close brackets is equals to I. So since nt over no is the finite survival rate, by definition, we have obtained. So log e, finite survival rate, is equals to the instantaneous mortality rate. So we thus obtain the following relationships for expressing the mortality rate. So the finite survival rate is equals to 1.0 minus the finite mortality rate. So log e into finite survival rate is equals to the instantaneous mortality rate. So the finite survival rate is equals to E, the instantaneous mortality rate. So the finite mortality rate is therefore equals to 1.0 minus E, instantaneous mortality rate. So why do we need to use the instantaneous rates? So the instantaneous rates are easier to deal with mathematically. For example, suppose you have the data on an insect population and you know that the mortality rate is 50% in the egg stage and 90% in the larval stage. How can you combine these mortalities? If they are expressed as finite mortality rates, we cannot add them because 50% loss plus 90% loss is obviously not 140% but 95% mortality rate. So if, however, the mortality rate is expressed as instantaneous rates, we can add them directly. So the egg stage is 50%, which is equal to 0.693. The larval stage is 90%, which is equal to minus 2.303. So combined loss is just 2.996. I know this looks a bit tricky, but it's very much straightforward. So we can convert back to finite mortality rate by the formula given earlier. So we just say the finite mortality rate is equal to 1.0 minus E, the instantaneous rate. So which is equal to 1.0 minus the exponential minus 2.996, which will give you 0.950. So and the combined mortality is seen to be 95%. So the idea of instantaneous rate can be explained most simply by the use of the compound interest. So suppose we have a population of 100 organisms increasing at a finite rate of 10% per year. So the population size at the end of year 1 will be as follows. So we have a population of 100 organisms increasing at a finite rate of 10%. So you need to change this 10% into real numbers. So we're going to say 100, which is the number that we begin with. Into here we have the rate 1 plus 1 over 10, which is equals to 110. So 1 divided by, so you supposed to work this out, and then you will get this number. You will understand it as we go on. And then at the second year, year 2, the population size will be as follows. So you're going to use the answer that you got for the end of year 1, which is equals to 110 into 1 plus 1 over 10, which will give you 120. At the end of the year 3, you do the same, and then you get your answer. For interest rate of 1 over m carried out n times, the population size will be as follows. So it's yn, which is just the amount at the end of the nth year, which is equals to y0, which is what you be having, the amount that is there at the start, into 1 plus 1 over m, which is 1 over m is just your rate, n is just the time. So this is what we need you to understand very carefully. So this example, you have 100 organisms to start with, And then you are told that they are increasing at a finite rate of 10% per year. 10% per year. So this is the time. So you're going to say 100. This is your Y0. So you are looking for the amount at the end of the first year. So it's 100 into 1 plus 1 over 10. And then which will give you 110. And then to find the number, this population size at the end of year 2, then you're going to use the answer that you got from at the end of the year 1, which is 110 into 1 plus 1 over 10, which will give you 121. And then to find the answer for the end of the year 3, you're going to use the answer that you got at the end of the year 2. So it's 121 into 1 plus 1 over. So this is your rate. This is your rate. Then if you want to see if that what you would do, what we have been saying is correct, just come here, put 100 and then put a 10 here, put a 1 here for the first year. And then as you go on, because now you have the time, again, Y0, what you have, put 100 into 1 plus 1 over 10. Now you want for the second year, then you're going to change here and put 2. And then you find the answer to be 121. And then you come again, you put 100 here, then into 1 plus 1 over 10, 3, and put the number to be 3. This is time, remember? And then you will find that is 133.1. But then if you are not using the formula, you are using the previous one, then you need to say 100 plus 1 over 10. And then you get the answer. The answer you get, you're going to use it for the second year. So this is what you need to understand very carefully. And then we're going to look at the age structure. So we're saying that organisms are born and they age until death. So the age structure is the distribution of the members of a population according to their age. So we usually calculate it as a proportion of the total population in each age class. So you just need to go through on this age structure section and then try to understand what is going on there.